Hello everyone. So this is our eighth lecture. We are kind of ending our course content with this lecture. Next week on Monday, I am hoping to keep a revision type of class. But since people are like losing interest, I'm saying if you are not going to join, if there is something important going on Monday for you, let me know. I'm thinking to put on a poll in Google Classroom. Let me quickly do it so that if the attendance is going to be low, like only one person is interested for Monday's class, then I'll cancel it and end the course with this class. Otherwise, if more, like 3, 4 of you are interested, then only I'll keep the class. All right. I'm hoping to give a quiz as well, but it's fine. This quiz is not mandatory from you guys or either for me. I just like to um, evaluate you so that I can evaluate myself. It's just to understand how much of this course is fruitful for us, me conducting it and you learning it. Is this the way of evaluating it? That's it. But again, quiz is optional. So I'll ask both the things, okay, in a poll after the class. If you're really interested, let me know through poll. Otherwise, I'll cancel it. So moving on, you have something to say? Anyone has something to say?
No, no, ma'am.
Okay, all right. So in today's class, we will understand another very popular and upcoming type of 3D bioprinting technique, that is light-based vat polymerization bioprinting. This is the fourth type, and we have already seen the past three types. How it is different from the extrusion one or the droplet one or light assisted bioprinting. Here we are using a vat, means a bath containing photo cross-linkable bio ink. Okay, and there is a stage, as you can see, and the stage can move in z direction, and the cross-linking of the polymer, it happens with the help of light, and this light is usually patterned. Okay, I'll explain to you later how it is patterned, but imagine that the light is patterned. So the CAD file that you give, it is digitally sliced in layers there, and each layer is basically mimicked as a pattern, as a light pattern. So wherever there are gaps in your CAD file, okay, or let's say in a layer of a CAD file, there the light will not form, the light will not fall. So that spaces will become dark. Whereas wherever there is you want material to be there, that space will become light. In this way, a black and white pattern is shown to light, and so when you give that black regions, the light will not fall through it, and where there is transparent regions, light will fall. So the light also get patterned in that way, and that patterned light falls over this vat. Okay, and a thin layer of photocross linkable biopolymer get cross-linked according to the patterned light, and then the stage moves, okay, uh, down. In this case, in this configuration, it will move down. Then a fresh layer of biopolymer comes, and simultaneously the light pattern for the next layer is shown over the polymer. So in this way, layer by layer, okay, printing happens using photocross linkable biopolymer. So very brief description of the technique. Now, in the upcoming slides, I'll go in much more detail. Okay. So, as I explained already, the same thing is written in the first point. that this technique relies on patterned light dose distribution to initiate localized chemical reactions of photoactivatable bioresins. As the bioresins react in response to light, this results in the formation of desired structures in 2D and 3D volumes. Okay, so when the patterned light falls over photocross-linkable biopolymer. Here, the term bioresin is used, and starting from this slide, I'll refer to biopolymer as bioresins. Okay, so don't confuse. These are just the same thing, and they are since they are photoactivatable, means they can form gel from solution in presence of light. That is why it is called resins. So most of the conventional resins are photocurable, means you have to shine some UV light and then they will cross link. That and since so they have taken the inspiration of resins into that in this paper. I'm using a paper, by the way, for making this content. So in that paper they have smartly used the term resins. Now this resins, since they are going to allow growth of cells and other biological components. So that is why it's termed bioresins. Okay, so don't confuse in the terms. It's just the polymer that we have talked about so far, which are photocurable, and they are called bioresins. So as I was saying, that as the light falls in a patterned way, the there are regions and spaces that receives some comparatively less light than others. Okay, so wherever there is high light intensity in the photocurable region, okay, there the photo reaction starts in a much more rapid way. Okay, and that basically starts cross-linking, and in this way you can make 2D patterns or 3D patterns. 3D patterns, basically we have to move the stage up and down in order to change the z-axis. Okay, so this technique basically came from the traditional stereolithography that was developed in 1986, and this technique was modified later for bioprinting almost 20 years later. The bioinks that are used are basically photocurable or photopolymerizable hydrogels, which are loaded with cells. Now there are various ways of doing this light-based vat polymerization bioprinting. It basically depends on how the light is falling and how the patterning is done. The structure of the light basically governs the way of patterning. You will see what I'm saying in the upcoming slides. So when we say point by point patterning, there is basically a laser point that is translated throughout the 3D volume of the bioresin. And under this, there are a few techniques that comes SLA and TPL. So here there is single photon laser lithography and there is two photon laser lithography. There is also multi-photon lithography as well. So basically how many lasers are used, okay, to create a single point of light that translates. So more number of lasers you use, you can ensure much more accuracy, okay, as well as the power density of the light also increases. So you will get much more sharper or much more localized chemical reaction going on in the vat, okay, and that will indeed help you get a much more sharper structure, okay, which much with much more defined features. Now the light can also fall in a layer by layer fashion, so that comes under layer by layer patterning. So very upcoming and a popular method uses this technique that is called digital light processing or DLP. Here, as I was saying in the first slide, you expose the bioresin with the patterned light. Okay, so that is basically how the DLP works. And lastly, it can be volumetric in nature. Okay, And for understanding volumetric, you have to realize that it is like there is a whole bath, okay, whole bath of the bioresin constrained within, let's say, a transparent jar or something. And the light that falls on the biopolymer, it is coming from all directions, okay. And each light pattern that is falling is the light is also patterned itself. So you see there is patterned light falling on the biopolymer from all directions. So the entire structure uh, gets made in few seconds, okay, because from all the directions, like if you rotate it, if we rotate the jar from all the possible directions, the structure is simultaneously being formed, and then it basically comes together in few seconds. That is called volumetric, okay. Here you use multiple lasers, or you use a holographic projection or something like that to create a 3D structure in few seconds. So briefly I explained to you. Now I'll go one by one to help you understand these modes of operation in much more detail. If you have any questions, let me know.
Ma'am, can you say it again how it works for once?
Which one? Volumetric one?
No, ma'am, there's a light-based vat polymerization.
Okay. So here what happens is there is a vat, if you understand, it's basically a bath, okay, or you just take a petri dish and pour some biopolymer in it. That creates the vat, okay. And this vat is kept on a stage like this, all right. And onto this uh like, what do you say, biopolymer, patterned light falls over it. So how do we make patterned light? When we load the CAD file into the machine, then that machine, either it can slice the CAD file into Z patterns by itself, or you can like just give the sliced file. So either way works depending on how powerful the machine is. So let's say the machine can slice the file on its own. And what it does, it creates some digital mask. Okay. So it will create some it will translate the Z patterns. So when you take, let's say, cylinder, okay, or you say a block, a cube block. So when you cut it in Z direction, what you'll get? You will get squares, right? So what it does, it translates each square into a digital mask. So the inside of the square, since you want the material to be there, it will be, in the digital mask, it will be converted into a transparent region, means white, completely white. Whereas outside, when you want no cross-linking, where you want no cross-linking, that region will become dark. In this way, if there are 100 slices, so 100 digital pattern will get created. That digital pattern is shown in front of the light source. So it's like you take a paper, you cut a hole of square shape, and then you put it in front of the light source. So what will happen? Light will be blocked, right, from everywhere, but the light will only pass through where there is a square. So in this way, the light that is going to fall on the, let's say, wall is going to be a square-like shape. So in this way, they make pattern light. And now this pattern light is allowed to be fall on the photocurable bioresin. So the sequence in which digital mask is being made and the sequence of movement of the Z platform, it is synchronized. Okay. So first layer gets shown simultaneously. The first layer, wherever the Z platform has to be, it is there. Ink is there over the platform, and you see cross-linking reaction happening only in the region where there is light coming. Okay. Rest of the vat is uncross-linked. So in this way, it continues layer by layer up to 100 layers. When you take out the structure, what you will see some liquid resins still left because the light did not fall on the unwanted regions. Light fall only on the regions where you wanted. And in this way, you will get um, like a cube. So this is like the mechanism for the layer by layer patterning. Okay, as I talked
I'm
about.
like
Yeah.
we pour the bio ink and then we will cut it from the tube.
No, you don't need to cut it since light is like falling only in the regions of the vat where you'd want, right?
Mm.
According to the digital pattern that you gave or CAD file that you gave, according to that, some digital masks are made and then light is patterned and you're getting light or you're supplying light or the machine is putting light in the vat only in the regions where you want. Rest of the region of the vat will remain uncross-linked. So it will
Then,
like wash out. It will just wash out. Rest of the polymer that is in liquid state, it will just wash out. You just have to take out and wash it and you will get the structure.
okay, ma'am.
Okay. It will be more clear, okay, as I discuss further. But I think you get the gist of it, means how the light is patterned here and why the vat is used. Did you like, if you have any questions in that aspect, let me know.
So what means polymer, solid polymer,
No, no, liquid polymer.
liquid polymer. Okay, on that we will place our what we place.
So this is a vat, okay, the blue
Okay.
region. It contains some photopolymerizable resin, okay, means a biopolymer which can be cross-linked or which can be converted to gel in presence of light. This is the blue region. And into this region, there is a platform. Okay. As the platform goes down, the liquid resin falls over it. Okay. Pattern light falls over it, and you get cross-linking only in the regions where you want it. Okay, where the light is falling, only there the cross-linking happens. The rest of the solution remains liquid only. So as the platform is moving down and down, more and more liquid will come, forming a very thin layer. Okay, and in this way, a structure gets formed from a first layer being at the bottom and the last layer being at the top. This is called top-down configuration. I'll come to this later, but this is how VAT is being used to supply the very large volume of resin, and light that is falling is patterned using mirrors. Okay, using digital mask. Okay, here there is digital mask that and mirror basically used to direct the light, but main thing is light and digital mask is there in this. In this way, layer by layer structure is being formed. Understood?
Yeah, got ma'am.
Mhm. But I'll explain in more details as well as we move forward. Okay. So when we compare the various modes of vat polymerization bioprinting technique with the another popular bioprinting, that is through extrusion, we see that extrusion on the minimum resolution of extrusion is around 100 micron, but all other types of vat polymerization bioprinting offers resolution much lower than the extrusion one. That means if you're interested in making very complicated structures like say vasculature network or some bones or liver with, like, when you're interested in mimicking the exact structural features with every nooks and crannies, then light-based technique, this vat polymerization bioprinting technique, can help you do that, because it can resolve very small features as well very nicely. Whereas in extrusion, you're limited by the nozzle or needle size that you're using, and the minimum needle size that comes is actually 100 micron. And also, if you go any size smaller than this, then you will risk in higher shear stress to the cell, and the cells will die. So for bioprinting, minimum resolution that you can go is up to 100 micron. The viscosity requirement is also different. So bioinks that are used for vat polymerization bioprinting, they are comparatively less viscous than the one used for extrusion. Okay. Now this is the values of storage modulus, means basically talks about the stiffness of the hydrogel. So you can again, you can work with hydrogels, or you can make hydrogel structures which are less stiff than the one that are possible through extrusion. The time basically depends on the minimum resolution that you're looking for. So for TPL, two photon lithography, since the minimum resolution that you can go up to 2 micron, so you will need more time. That makes sense. Okay, but then for VAM, a volumetric additive manufacturing, I'll talk about it a bit later. Here the resolution is around 40 micron, bit higher than three ones, all the three. But since here the light is falling in volumetric world, since the light is falling in all the direction, from all the directions on the vat, that is why the structure is getting formed simultaneously from all directions. You don't have to move any platform in Z direction or anything like that. Okay. So that structure, in like how big of a structure, you don't have to worry about it. Okay. So it can make very big centimeter size structures in few seconds. So if you now compare extrusion with light polymerization bioprinting, what you see that light polymerization bioprinting, vat polymerization bioprinting, it offers much more control over structural complexity, means you can create very difficult to mimic structures using this. Okay, you can create nerve structures, blood vessel structure, lymphatic vessel structures. Then people can also create spine, bone structures, which are very complicated, right? You can take the help of medical images and then try to mimic the exact features using vat polymerization bioprinting. High resolution, definitely it has high resolution because light that is falling, it is very, uh, the way it is falling and the patterning and all is very much precise. Okay, localized chemical reaction happens within the vat. So only the regions where the light is falling, there only the printing happens. Means there only the structure will get formed. The liquid bioresin will get converted to gel only in those regions where there is light falling, and the fabrication rates are faster. So, like, it's, I would say it's comparable if you uh, compare with extrusion, but yeah, relatively it is faster for a few modes, and then somewhat it is comparable for a few modes. But yeah, main 2 points that are to note is that you can create very good resolution structures, and second, you can create very complex structures as well. So we talked about point by point patterning of the polymer. So under that, there comes a technique that is called two photon lithography, or TPL. So as you can see, this is the vat. This vat contains a red colored polymer solution. Okay. And this two photon laser, it can move anywhere, XYZ, anywhere within the vat. And wherever it is moving, the cross-linking happens there and you get a solid structure. So why it is called two photon? You see, one laser is coming from top, another laser is coming from bottom. They are basically meeting at one point. Okay? So you do that in order to make sure that there is more accuracy. Okay. And you also increase the power density of light at that meeting point. And wherever the point meets, there the cross-linking happens. Okay. This is the representation of two photon lithography. So here what happens? There is point by point scanning, first of all, and there is something called raster scanning that happens. So what is raster scanning? It is it also happens in our cameras as well. Okay, CMOS sensors that are used in cameras, they're for generating each pixel of the photo. Scanning happens in like this. The light is scanned in that way, and you get pixel by pixel resolution of a photo. That is called raster scanning. So if you look at this image that I've given at the bottom, here if you see in 2D plane, XY plane, each point is scanned, okay, and then moves forward. Another point, so in this way, one row gets scanned completely, then it will move like this, and then another row like this, another row like this. This is called point by point. So each cell here represent one point within a 3D structure. Okay, and as I said, the CAD file will get sliced. So each sliced version is basically one XY plane, and in one XY plane, one cell, you can say imagine from here, one cell is basically one portion, one very small portion of the sliced CAD file. So here the laser will fall first, and then it will fall in here. So in this way it will move, and it will complete the entire slice section, and then it will move forward with the next sliced section. So in this way it will scan, the laser will move throughout the vat, okay, and create a 3D structure. Now this approach, it provides very efficient photo reactions because the power densities of the laser light is very high. So you get very efficient way of photo reactions happening. But since the power densities are quite large, it can result in very low cell viability. And it's a very slow process for building large volumes. Okay. Any questions in understanding how this technique works?
No, ma'am.
Mhm. Okay. So people have used this technique to create constructs which are limited to, let's say, hundreds of microns to few millimeters. So since this technique takes a lot of time, okay, as it is going to do raster scanning like this, like row by row. So possible, like physically possible way of making or using this technique make work is by making structures that are very small, like few millimeters. But if you look at it, it holds a very great promise to create micro level tissue models, then cell and drug delivery systems, which are microstructured, like the microneedle arrays and all. And this technique is generally very expensive because you're using laser systems and all. So that increases the cost of this technique. As you can see in the photo that I've given in the slide, People have attempted to create very complicated structures using two photon lithography. So image one is basically an image of blood vessels, and where the cells are basically seated inside it, as you can see, and um, they have formed very good lining inside the blood vessels. In figure B, here the blood vessel is made in form of a glomerulus. So glomerulus is a part of a kidney function where the blood comes. Okay, here the surface area is very high, and then it gets filtered out. So that structure they have tried to mimic through this technique. You can see very good resolution here. Then in C, here they have tried to pattern growth factors in order to guide the growth of axons. So these are basically the colored images are basically the nerve cells, the blue regions of the cell nucleus, okay, and the green regions are basically showing how the growth of axon is happening from the cell body of the nerve, and like these are the guides that they have made as well. Some you can see thick channels like structures that they have made guides, and they have tried to ensure that the axon grows within these guides. Okay, so in this way they have used this very powerful technique to create very complicated structures which are found in our body. Okay, any questions?
No, ma'am.
Okay, then another technique, another way of using this vat polymerization technique is by doing layer by layer pattern. All right, so that is called digital light processing or DLP. Now this is the machine that's very expensive again, and also highly used nowadays for bioprinting. Okay, it is not as expensive as the TPL one, but it is expensive than a extrusion system, but it is widely used nowadays because here the toxicity to the cells due to light is much less. So here what happens, as I told you, you get light, projected light, and that light basically comes out as a pattern, okay, digital pattern. It's reflected and allowed to fall directly on the vat. Now, the first image that I showed you was a top-down approach. This image that I'm showing here is a bottom-up approach. Here, the first layer is formed in the end, whereas the last layer is formed at the beginning. Okay, so this is another representation of the same system that I'm explaining here. So this is the DMD or digital micromirror array, all right. So this DMD is, it can create a mask like this. This is called digital mask. So wherever there is a dark, the light will not fall, okay. And wherever there is transparent region, white regions, the light will fall there. Okay, this is the CAD file, let's say, and that CAD file is sliced like this, and each layer or each sliced version is converted into digital mask like this. Now this DM, so light is falling here. This is the UV light. It falls over digital mask, and then it basically falls on this pattern, and the light comes out as a point of after reflecting. The light that comes out after reflecting from this DMD, it reflects out in the form of this pattern. Okay. Then there are lenses placed to make sure uh, to minimize the entry of scattered light. Only focused light will fall, okay, on the polymer, and in this way polymer uh, gets patterned according to this mask, and this then layer by layer you get a 3D structure like this. Okay, is it clear now?
Yeah, ma'am.
Hmm? Okay. So, just what I explained, that is only written here. A single plane of light can be projected at once to enable simultaneous photocross-linking of the desired pattern in that layer, followed by layer construction leading to 3D bar printer structure. Okay. They use what light emitting diode arrays that directly emit patterned light via LCD screens that form digital mask in front of the light source to achieve patterned light or they also use digital micromirror array devices that reflect the incident light to build platforms. So what they do, how they display the digital mask in front of the light, there is an LCD screen, okay, that forms this pattern. Let's say this forms this pattern, and the light source is like reflected using mirrors to fall directly on this pattern. And as after since the LCD screen is transparent, right? So it just pass through that uh, this pattern, and you get the pattern light directly falling on the build platform. People use non-laser or low power lasers. So that makes the technique much more cost efficient, but the resolution is reduced. That is around 50 to 100 micron range. The resolution is possible. Okay. Now these are some amazing work that people have done using this technique. Basically, it is possible because this technique is much more accessible to people or to research labs of different budgets. So that is why it is much more appreciated nowadays for bioprinting. So this figure sorry, B figure BA is like one of the very um, recognized work here at Rice University. There is a professor here at Rice whose lab did this, and then he was like so much appreciated that that guy earned a lot of money. He was given a lot of money to leave his job as a professor and then join a very big 3D printing company. And now he is a director of the bioprinting sector of that company. Okay, so this is because of this work. They have basically made a structure of alveoli in the that is the unit structure of the lung where gas exchange can happen and in presence of blood. So they have made ducts like this for the oxygen rich, so deoxygenated blood to come in, and then there will be alveoli cells, lung cells, where the air will pass through. So air will pass through, and you will get oxygenated blood out. So that model they have made using this DLP technique. Okay. Then here in B, people have made very big structures like structure of an actual sized hand. And inside these structures, they have made perfusible channels. Why perfusible channels is important? It is because if you're making a very big structure with cells inside it, then the cells which are basically in the bulk, they will face a hypoxia or nutrient starvation if you just keep it dipped inside a media bath, then enough diffusion of media will not take place towards the core of the structure. And that is why the cells in the core they can start dying. Okay. So to prevent that, we need to provide paths through channels. So that channels they made inside this big structure. So this is the structure of a liver, actual size liver. And inside the liver, they made channels like this. Okay. So these are all very cool work that people are doing with this technique. And the same principle, this is the same principle of the machine. Like, and given what you want to achieve, what's your target, people have made very good structures with high resolution. Any questions?
What was the company's name, ma'am?
It's called volumetric. He and his PhD student made that company. Then it was based on this 3D bioprinting only. Then it was procured by a very big US company that is called 3D Systems. If you search, 3D Systems is a very, very big 3D printing company. So they procured that company for hundreds of dollars and like for millions of dollars, actually, not hundreds, millions of dollars. And now that guy is like set for life. Yeah.
Okay.
Yeah. I didn't knew about him until I came here. His work is apparently very much recognized in my department that I worked here. So yeah.
Oh, while he done his work, this work in India, ma'am.
No, no, he is a professor. He was a professor at Rice, not in India.
Where, ma'am? Rice.
Rice University,
Okay, ma'am.
Houston. Yeah. Talking about Rice University. A professor who was from. So this work was published in Science Journal. Okay. No questions regarding technique? Any questions regarding this technique?
No, ma'am.
Okay. Next, there is the volumetric additive manufacturing or VAM. Here, what happens? There's a jar kind of a thing that can rotate. Okay. And as it is rotating, a sequence of projected sequence of pattern light, it falls on the vat. And in few seconds, a very big structure like this, let's say, gets made. Okay. So the mechanism of creating pattern light, I think you have understood by now. The same mechanism is being used here. Only difference is how the vat is kept. Okay. In the DLP one, the vat was like able to move only in Z direction, right? Because it is kept on a platform and that platform is it is motorized, so it can move in Z direction. Here, the vat is kept on a platform which can rotate. Okay. And this rotation and the falling of pattern light, these two are synchronized. So as it is rotating, pattern light for that particular region is projected and a specific and photocross linking will happen inside the vat to create a 3D structure like this. So here we are using multiple planar light patterns, right? Which are produced either from a laser light or from a non-coherent light source. And they are all subsequently projected across the entire volume of the vat. These projections, they create an anisotropic light dose distribution that exceeds the polymerization threshold of the bioink only in the object's intended geometry. So the light patterns that are falling, they are basically optimized to create the light dose, okay, needed to cross the threshold value for the photopolymerization reaction to happen. So if you don't cross the threshold, then the reaction won't happen and then you won't get a 3D structure. Then VAM is currently being performed either using a single light source where it is projected onto a rotating vat. Okay. Yeah. So this is an example of tomographic bioprinting, or you can modify it by using a static vat, but multiple light sources are used. Means the vat is static, but the light is coming from multiple directions. That is called holographic printing, or you can also use movable light sheets, okay, that intersects orthogonally with DLP projections to trigger vat polymerization owing to uniquely designed photo initiators. This is called light sheet 3D printing. This in this light sheet 3D printing, what happens? The DLP projections means the projection that we just talked about in horizontal direction, and there is a sheet of light that will move like this, okay, and cross the horizontal projections, okay, and so wherever the crossing happens, okay, there only the intensity of light dose is high for cross-linking to happen and you get a 3D structure. Got it? So let's say this is the stage, all right? If you can see my hand, this is the stage onto which vat is kept. And the sliced sliced light, or you can say pattern light, it falls on, let's say, first layer. Okay, so the but the intensity of light that is falling is not enough for cross-linking. So then a moving light sheet will cross this region like this. So wherever the crossing happens, the light intensity is basically higher there and the structure gets formed. Then it moves a little bit up. Again, a sheet of light gets crossed like this, then like this, like crossed like this. So in this way, a 3D structure will get formed um, in this technique. Okay, got it? Got the three types? How the VAM or the volumetric additive manufacturing is modified?
Yes, ma'am. To a level, ma'am.
Yeah. Then VAM process, as VAM process address the whole volume at once, it can manufacture at a very high rates. That is less than 20 seconds it needs to generate cubic centimeter size structures, which is like very fast rate, right? The rate is much faster than other vat polymerization strategies. Okay. So here are some examples where people have used this technique for creating very complicated structures. Like in the first image here, in where my laser pointer is, they have tried to create the structure of mouse pulmonary artery. So you can see this is the CAD file and this is the actual structure that they have made using tomographic printing. So in tomographic printing, the vat is rotating and light is projected in one direction. Okay, that is tomographic printing. This is tomographic printing. Okay. The second thing, the second image, they have tried to create the structure of trabecular bone. This is the structure CAD file, and this is the CAD file, and this is the actual structure. And inside the trabecular bone, they've seeded mesenchymal stem cells. So you can see the cells also here, but the image is very dark. But yeah, it is there. Okay. Then in C, they have used C2C myoblast loaded a complex model. So they have basically created a very complex architecture like a helix, something like that. I'm not able to make it out. So this is a structure in which they have certain myoblast cells. So here you can see the cell nuclei that is stained blue very nicely, and there are myosin filaments that are coming out that is stained in red inside the structure. And you can see pattern like a spring-like pattern that is being shown with the myocytes. Okay. So these are the three main common types of light-based vat polymerization bioprinting. If you have any questions, let me know, because now I'll move forward with that will help you guys understand how to choose a bioresin for this technique. Okay, what properties should it have? What are the bioresins currently being used? So we'll discuss that in a minute. But if you have any questions regarding the three techniques that we discussed, let me know.
Ma'am, no questions, ma'am.
Okay. Now, what kind of bioresins people are using? The first point that to note is the choice of bioresin basically depends on what type of method that you're going to use. Okay. For example, if you use DLP, digital light processing type of a technique where you're using layer by layer patterning, it uses bioresin which are of low viscosity. It should be able to flow well. Whereas for TPL, two photon lithography or VAM, volumetric additive manufacturing, here you require relatively more viscous bioink resin or bioink or bioresin. So typical bioresins that are used currently, they are either PEG, polyethylene glycol, gelatin, or hyaluronic acid. Now they are modified, their backbone is modified by adding some photoactivatable side chains. So these are usually acryloyls, methacryloyls. Okay. So in presence of these side chains, there will be chain polymerization reaction, or people also add some thiols or norbornenes. So that will allow step growth polymerization reaction. Okay. So also one thing to note is what type the kinetics of your cross-linking reaction. It should proceed at an adequate rate. Okay. It should not be too slow, it should not be too fast. It should be like optimized, because we need this optimization to happen because it can cause unnecessary sedimentation of the cells. And what happens if your process of cross-linking reaction, that if it takes longer period for the cross-linking reaction to complete, then the cells will not be fixed in that particular layer. You will see inhomogeneous distribution of the cells. The cells, since they are like acting in the gravity, so they will start to settle down. And each layer of the 3D printed structure won't get the right amount of cells. So the reaction kinetics, it should happen at an adequate rate to prevent the sedimentation of cells. Okay, but it should also be mild enough in order to ensure highest possible cell viability. So it cannot be very fast, because if it's very fast, then there will be more release of free radicals, and those free radicals are basically can kill the cells. But if it happens at a very slow rate, then enough free radicals are not being generated, and then the reaction is slow, so cells will settle down. So we need to balance this. People use light sources like UV 365 nanometer is UV light, or visible light like 405, and they use photoinitiators like uh, IGRA-Cure 2959, and then they use LAP, that is lithium phenyl 246 trimethyl benzoyl phosphonate. Okay. And photoinitiators are often complemented with some photoabsorbers as well. And the concentrations of photoinitiators and photoabsorbers, they are very less. They're in the order of millimolars or less when you compare it with the reaction, with the concentration of reactive functional groups. Reactive functional groups basically mean the acryls and methacryls and thiols and norborins. So when you compare the concentration of photoinitiators and photoabsorbers, they are very less in comparison to these reactive groups. So these reactive groups are like hundreds of times higher in concentration in comparison to this photoinitiator. So you need very, very little amount of photoinitiator and photoabsorbers in your bioink formulation. So if I can summarize what all things that you need to make the bioresin, first you need a functionally modified polymer. Okay, like PEG with methacryl or gelatin with methacryl, then you need to add the cells at some cell density. Then you also need to add photoinitiators to initiate the photo cross-linking reaction. And lastly, you also need some photoabsorbers. The role of photoabsorbers is to maximize the resolution. Okay, how it does it, I'll come to it later. And then people also add some oxygen inhibiting molecules that also helps in maximizing the resolution. Then people also add some viscosity modifiers like glycerol or something like that in order to make sure the viscosity of the entire bio ink or bioresin is suitable for the process. That's it. Like around you need 4 or 5 things. So making bioresin for this application is that makes the job of making bioresin for this application a bit more complicated because you have to work with so many different things. Like you have to optimize for each of the concentrations and how these concentrations are compatible with each other. So it takes years to make one bioresin for this particular technique. In this box, here they have given the names of the hydrogels that are being used. Okay, then photo cross-linking chemistries, what type of photo cross-linking chemistries are generally used. Then what type of small molecule additives are used, like photoinitiators are added, then absorbers, photo absorbers are added, oxygen inhibitors are added. Okay, then some resins do not have enough refractive index for the optical property optical property of the bioresin is poor, so they add some viscosity refractive index modifiers. Okay, then yeah, so these are things that are added, and there is some special consideration as well. So when I'll share the slides with you guys, you can have a look. Okay, to this box. It is also there in the reference paper that I'm using to um, teach the content for this topic. So now the photopolymerizable reactions. Okay, so it can be of two types that I briefly mentioned. One is chain polymerization reaction, another is step growth. So each has its advantages and disadvantages. So chain polymerization reaction means there is like first, in presence of light, the photoinitiator will get activated. It will release some free radicals, and those free radicals goes and targets the, let's say, methyl group or acryl group. Okay, and then new free radicals get generated, and in this way each of the newly generated free radical targets the unreacted groups. So there is one initiation step, propagation step, and lastly termination step. Okay, so in this way chain wise the reaction proceeds. So here what happens, through generation of free radicals the reaction proceeds. For initiating this chain polymerization reaction, you need a very little amount of photoinitiator, and this reaction can cause conversion from liquid to solid hydrogel, like at a very low conversion rate, means around less than 2%. Okay, if less than 2% of the reactive groups are targeted, then the structure starts getting formed. But it is very sensitive to oxygen inhibition. If there is lot of oxygen inside your bioresin, okay, then what happens? The free radicals won't be able to target only the reactive groups. The oxygens will also get targeted, and it basically then hampers the photocross linking reaction that is happening. So, which is why it can often result in inhomogeneous, brittle networks. Okay. By comparison, the step growth polymerization reaction, here what happens? Step by step reaction proceeds. So the photoinitiator in presence of light, it does not instantly generates the free radicals. S- like there is first two compounds, then like so whole other chemistry. But in the polymer here, what happens? It, first, the free radical targets two nearby reactive groups. They are converted to their free radical form. They react with each other. Okay, that's it. Free radical khatam. There is no more free radical. And then again, another molecule of photoinitiator that gets activated to generate free radical. Again, it targets the unreacted groups. They are formed a free radical, and then network is formed. So there is a step by step method. Here, the free radicals which are left at the end of the reaction are much less. So, which is why if you need the polymer to convert from solution to gel state, you need a large number of conversions to happen to get a gel state. Okay. And this reaction is much more tolerant to oxygen presence. So oxygen cannot really do a lot of harm here. So the 3D network, 3D hydrogen network that you get is much more homogeneous and tougher. Okay. Any questions? If I'm going fast at any point of time, let me know. Okay. I'll pause and help you understand any doubt that you have in much more better way. I'll try my best. Any questions?
No, ma'am.
No? All right. So for improving the feature resolution, let's say for chain polymerization reaction, people also combine photoinitiators with free radical scavengers like TEMPO. Okay? It stands for 2266 tetramethyl piperidine 1-yle. Okay, so it can basically scavenge out or basically hunt out the oxygen that might be present within the vat. So in that way, oxygens cannot interfere with the photoinitiator activity, and then it can improve the rate of photocross-linking reaction and improve the resolution in that way. But it overall slows down the reaction rates as well. So there is a balance between how much of tempo is beneficial and how much it can harm the reaction. Then people also use photoabsorbers. So what are these photoabsorbers? They're usually a non-reactive molecule, means they will not participate in the photocross-linking reaction. And they usually contain some chromophores, pigments like colored pigments, okay, that can absorb light in the same range as the initiator. So what it will do, it will then reduce the light penetration depth, so preventing overcuring and improve the feature resolution. So these are basically some dyes. So tartrazine, this is a food dye, yellow colored food dye that you, and this dye is very heavily used for making your food yellow, like nachos or Doritos and all those chips and all that are very heavily colored. So this type of color tartrazine is used in those. Curcumin is also a type of curcumin is a dye that is found in haldi. So turmeric gets its color from curcumin. So these kind of chromophoric dyes, they are used as photoabsorbers. Why? Because they can absorb the light that is falling on the vat of the same wavelength as the photoinitiator. So what it will do, it will not allow scattered light. So light will definitely scatter inside the vat. So it will not allow the scattered light to cause photo cross-linking reaction at the region where it is not needed. Okay. It will absorb the scattered excess light from the rest of the region. So it will then improve the resolution of the final structure. And after printing, it is very easy to remove them as well, because since they are not going to get react or not to, it's not going to react with the biopolymer itself. It's just like scattered as molecules, as free molecules inside the 3D structure. So we can just take the 3D structure out from the vat, put it in a washing solution like PBS or water or something like that, and all the dye will wash out. So it's not going to even harm the cells, because they are mostly food dyes and they are already certified for safety for us. So they're very good in making sure that the resolution is very good in this technique. Okay. Now, also one more thing, cells, they are transparent. If you see, because you can observe cells under microscope, it is because light can pass through it. And also there are some tiny structures inside the cell that can scatter light. So they are inherently a light scattering structures. So as I said, the light where it will fall unnecessarily, the light will get scattered. Why? Because of the presence of cells. And that scattered light can get absorbed by photoabsorbers and prevent unnecessary overcuring at the regions where you don't want. The cells are inherently light scattering, and they can sediment as well. Okay. If the kinetics of the photoreaction is very slow, then it can sediment. And this sedimentation of the cell, it can lead to inhomogeneity in the distribution of the cell inside the bioprinter structure. Thus, people often use some high molecular weight photopolymerizable precursors or some additives like colloidal silica to alter the bioresin viscosity and thus reduce sedimentation. So, if you make the solution much more viscous, it will has a less tendency to flow, and the cells can, get trapped at some 3D space inside the cell, inside the vat. So that will uh, prevent inhomogeneity in the bioprinter structure, or inhomogeneity in cell distribution inside the bioprinter structure. Okay. So any questions so far?
And can we use only percoil or the alteration for the viscosity, ma'am? Is there anything we can use?
Yes, you can use percoil to alter the viscosity. Yes.
No, other than that.
Oh, other than that, I have seen or I've read people using glycerol.
Okay, ma'am.
Mhm. Okay. So now now that you have understood how to choose a bioresin, what properties to look for, what all things that goes into the bio ink that is being used for this particular technique, should also understand there are various ways of doing the light-based vat polymerization technique. So here, these are the variations of DLP mode. Okay. I'll discuss four variations, but all the four variations are possible for the DLP mode. Why I'm showing variations for DLP mode? Because it is much more being used. All right. So more and more research is available for DLP in comparison to other light-based vat polymerization bioprinting. Okay. So in DLP, a digital light processing way of doing vat polymerization bioprinting, here what happens, you can either print the structure uh, with the bottom layer printing first and the first layer printing at the end, that is bottom of configuration, means the structure gets printed inverted, all right? or you can print it in a top-down configuration where the first layer is at the bottom of the platform and the top layer is at the tip of the platform. So the structure that gets formed is upright. Okay. So depending on what configuration you're using, the direction of light will be directly opposite to it, opposite to the building plate. So if the building plate at the top, then the light will get projected from the bottom through an LCD screen. If the building plate is at the bottom, then the light will come from the top. And people also use this kind of a vat here as well. Okay. So there are advantage and disadvantage in using either of this configuration. So let's understand one by one. In bottom-up configuration, people pull the construct up, up in upwards direction with the preceding layer is cross-linked. So as the preceding layer gets cross-linked, a tiny space, very thin, tiny space will get exposed between the preceding layer and the bottom of the vat containing the liquid resin. And that thin space get filled up with the liquid resin, which is very thin layer. Okay. And that thin layer gets, cross-linked for the next layer to be formed. And that layer gets, mixed with the material, overall structure. So in this way, layer by layer, the structure is made. This technique ensures you can print a structure very well using very small volume of bioresin. Minimum volume of bioresin, you can print it. There is sufficient mechanical stiffness available in this structure. And if it is not, if the structure is not stiff enough, then gravity will, pull the structure out and you will see the structure will crumble, or layers will not adhere with each other well. So you need to make sure use a bioresin formulation that gives sufficient mechanical stiffness to your structure so that it can overcome the gravitational force that is going to pull the structure down. You can do printing with single material, or you can do printing with multi-material as well. So I'll come to that multi-material part in the next slide. In the top-down configuration, here what happens, there is a big vat, okay, and it's big enough to accommodate a large volume of the bio ink as well as the platform. So it has to be big, it has to be deep enough for the structure and the build plate to be accommodated. So for making even a very small structure, let's say millimeter size structure, you have to use very large volume of the bioresin. So that will result in wastage of the bioresin. Okay. So as the build plate move down, okay, very thin layer of um, bioresin gets exposed over the structure and onto which a projected light falls, and a layer gets cross-linked. Okay, and it contributes to the overall making of the 3D structure. Here you also need to make sure that the surface tension of the liquid, so at the top layer of the bioresin liquid, surface tension is high, right? So it can disrupt the smoothness of the liquid bioresin. So one needs to optimize for the surface tension properties of the bioresin as well in order to make sure the correct structure is formed and with high resolution. All right. So these are two ways of doing DLP. One is bottom up, where the structure is formed in an inverted fashion. This is top down, where the structure is formed upright. Okay. And depending on where is the build plate, you will find the light projecting from the opposite direction. Then you can also do multi-material bioprinting, although it is not very common. but people are coming up with multi-material bioprinting as well. Here, what happens? You can either use multiple machines or you can use multiple vats, okay, at a time, because there can be only one vat and the vat can contain one material at a time. Usually, like that's what our mind says. But an exception to that is also available that I'll discuss next. So in one vat, there is a single material, and you can change the vats in order to add multiple material to your 3D printer structure, or you can move your build plate, okay, to get exposed to multiple material from a different machines. You can do that as well. Or what you can do, you can use single vat, and that vat is, let's say, connected to some microfluidic channels. And through that microfluidic channels, you can fill the vat with different materials whenever you need it during the printing process. And then you remove the material that you don't need. So using channels and using syringe pumps or make an automated syringe pumps and all, you can supply the liquid bioresin 1, okay, that you need for the, let's say, first layer, then remove bioresin 1, then through channels you supply bioresin 2, again fill the vat and then do the printing. Or you can do the entire thing that which I just said manually. You can manually fill the vat with bioresin 1 with your pipette or something like that, and then you pause the printing, remove the ink, remove bioresin 1, then add Bioresin 2 manually, then start the printing again. So you can do it manually as well. So back and forth you can do. And in this process, you can either stop the printing or pause the printing to wash the 3D printed structures and then start printing with the new resin. Okay, so you can incorporate a washing step in between changing the type of the vat. um, or in between changing the type of the bioresin, you can do that. So in this way, you can do multi-material bioprinting as well. Or what you can do, you can use multiple wavelength light. You can keep one bioresin or one vat with containing multiple types of bioresin. Okay, you can have one vat with multiple type of bioresins, and each bioresin you have to make sure that it can cross-link at different wavelengths of light. So you are basically using multiple light sources in, or you are using, let's say, one light source and multiple polarizers in order to segregate light depending on what wavelength you want. Okay, and these multiple wavelengths of light, they are falling on the vat at the same time, but the composition of the bioresin is is not one. It is not homogeneous. There is many more bioresins available. Let's say two are available, and let's say bioresin 1 corresponds to wavelength 365 and bioresin 2 corresponds to wavelength 405 nanometer, and only in presence of that wavelength the cross-linking will happen. So you can basically uh, modulate what kind of digital mask is getting projected from which type of light wavelength. And depending on that, you can create a structure which is inhomogeneous, means you will find few regions made with bioresin 1 and few regions made with bioresin 2. So that is another way that you can do multi-material bioprinting with multiple wavelength of light, okay, in this DLP kind of a system. So that's it from my side. So I use this Nature Reviews paper um, to describe the content for today's class. I'll share this paper with you all. All right. You can go through it in much more detail. There are other stuff mentioned as well. but for the sake of keeping this course easy, I omitted some of those. But if you're interested, you can go through the paper and you will get much more deeper insight. This is a very nice paper. It was published in last year, 2023. It was published, so very recent one as well. Because as I told you, this technique is becoming much more popular in the bioprinting community, and more labs are like getting funds to secure a machine like this, and they are like doing cutting edge research in this technique. So that's why you get a recent paper in this. So go through it, um, go through the notes as well, and if any questions at any point of time, let me know.
Yeah, okay, ma'am.
Okay. Any questions in understanding any point or any slide during the lecture?
Not exactly, ma'am.
Not exactly. It's fine. It's fine. Yeah. I hope the I was not very fast. I hope I was able to, catch your attention. I hope you didn't drift off in between. Was I too fast?
Yes, ma'am. Compared to your usual, it is fast.
It was fast. Oh, it's fine. Let me know if you want me to repeat some stuff.
No, no from my side.
No? Okay. What about you, Guru? Okay. Um, Sorry, can you repeat what you said? It was very mild. Your voice was very mild.
Ma'am,
Yeah. Do you have any questions?
no, ma'am.
No? Okay. Um, you can go through my recording again. I'll share with the recording with you to catch some points that you may not have understood, and go through the notes as well, which I've made here. That is also very much self-explanatory. But I think after that you will get to understand a bit more. Okay. So I'll put up a poll in classroom to understand whether I should have the class on Monday or not. But what are your opinions like? Let me first stop the recording.